Hey everyone, Professor Davis here, and we are going to be looking at microbial growth. So when we look at microbial growth, we're going to be looking first at some physical growth requirements, and then we're going to get into some chemical growth requirements. So the first thing we want to look at are the physical growth requirements that include temperature, pH, and osmotic pressure. And we're going to go through each of them, and we're going to start with temperature first. So temperature, we do see that bacteria are going to have optimal temperatures to grow. Temperatures which the bacteria has the shortest generation time, like it reproduces the quickest during that time. There's three basic categories based on preferred temperature ranges. The range can spread of about 30 degrees, okay? Growth is not optimal at the end of the ranges, and we talked a little bit about this in the previous video on how the enzymes and stuff that help with growth can be affected by temperature. And if they denature, then that causes metabolic issues, which then ultimately affects growth. The first group we want to look at are the psychrophils, and they're the cold-loving bacteria. They are going to be the ones who are at the bottom of the Arctic and Antarctica oceans, okay? So when we look at this, they're going to be where it's super, super cold. Now, we don't really mess with a lot of those because the temperatures around us, for the most part, don't get that cold. And so we are going to actually be more concerned with what we call the psychrotrophs, the ones who like the cooler temperatures, especially what we see with the refrigerator. So these are the microorganisms that actually can cause food spoilage in our refrigerators, and they can grow at room temperature, but they also like things that are a little bit cooler. One example of this is Listeria. Listeria prefers temperatures between 30 degrees Celsius and 47 degrees Celsius, but they can grow in the range from 1 to 45 degrees Celsius. So they can grow where it's really cold. Listeria is normally linked with like lunch meat, processed lunch meat. Okay, so these are the ones who like the cold. We then have the mesophils. The mesophils are shown here in kind of the yellow color. They like the moderate temperatures. This is kind of the most common. These guys are going to grow at best between 25 and 40 degrees Celsius. They have rapid growth during that time, and some of them also produce toxins. We see that most bacteria are mesophils, and almost all the pathogens that make us sick are mesophils because our body temperature is in this range, because our body temperature is 37 degrees Celsius. This is also the most common food spoilage organisms, okay, the ones that fall in the mesophil range. The thermophils are the next one. These are the heat-loving. These are found in like things like hot springs where temperatures are actually very, very high. If contaminant is in a dairy product, it could potentially survive pasteurization. We talked about what pasteurization was back in one of the first unit videos, but we do see that a lot of times these guys are going to live in those extreme heats. They don't grow well at body temperature, so most of them are not pathogenic. And so because of that, for food spoilage, they're not really a public health issue. All right, because they like the more extreme heat temperatures. And you can see that here in the chart for red. The next thing we want to look at that's a physical kind of characteristic of growth is going to be the pH. Now, optimal pH is normally between 6.5 and 7.5. Most microorganisms, including bacteria, like this range. Now, sometimes your fungus, like your yeast and molds, they like it to be a little more acidic, and they can normally grow between the pH of 5 and 6. We do see that there are some acidophils. Acidophils are going to be your Archaea group. These guys like to tolerate acidic conditions, and you can see that in the blue. So just for your information, we do see that acidity below pH of 4 is used to preserve food. We see this by like when we pickle things. We put them into a substance with vinegar and other things in order to bring the pH down, and we pickle that. They do need buffers in artificial media because some of the byproducts, a lot of the byproducts of metabolism are acids. And so if the acids accumulate on our plates that we're trying to grow bacteria on, it will then kill them because a lot of them can't live in that acidic condition. So we do see that those plates will have buffers present in order to take care of those acids being produced. If we look at Helicobacter pylori, this is also known as H. pylori. This is one of the most aggressive types of bacteria that are found in the stomach. They do cause potentially ulcers, gastric ulcers in your stomach. They are actually not acidophils. They don't actually like the acid, which is crazy because your stomach is full of it. But what they do is they embed into the mucous membrane, and that mucus layer that protects your cells then protects it. And it then can grow and flourish in that environment. It's also why it's super hard to treat H. pylori type of infections with antibiotics. The next physical type of characteristic for growth is going to be osmotic pressure. Osmotic pressure is talking about the water concentration. Cells are about 80 to 90% water. So how much water is found outside of the cell and what concentration is going to determine what happens to that cell. So if you'll recall, when we were talking back about cells in the Unit 1 videos, we saw that there were hypertonic solutions, hypotonic solutions, and isotonic solutions. Well, hypertonic environments or solutions are going to cause a lot of these cells to do plasmolysis, meaning cell shrinkage. The water is going to move out of the cells, causing them to shrivel up and shrink. Now, there is a term in the Archaea group called the halophils who love salt. Salt is what makes a lot of times things hypertonic. They actually really like it, and they can live in like the Great Salt Lake or even in the Dead Sea. But most things cannot survive in those high salt concentrations. So this is why salt is used a lot of times to preserve food. Before people had refrigerators and things like that, they would normally take like their meat and stuff and they would rub salt all over it to help preserve it. And this would help keep bacteria and fungus from growing on it. Augers tend to have a 1.5% higher concentration, which creates some hyperosmotic conditions. So we have to be a little bit careful, but we do see water as a byproduct that comes out of these cells a lot of times on the plate. So there'll be condensation on your plates when they grow. So plasmolysis does inhibit cell growth, which can be a bad thing. Here's an example here of some bacteria that are haliotolerant, meaning they do well or okay on salt augers. Example here is a Staph aureus. It can be used, this type of plate with the salt concentration can be used to ID organisms, at least the ones who like to grow on the salt content versus ones who do not. Another one is the hypotonic environments. This may cause osmotic lysis. In the hypotonic, the water goes into the cells and they will explode and burst. And so they're not going to be successful either if they're in a hypotonic solution. So what are factors used to reduce food spoilage? If we're looking at these physical kind of environmental type of characteristics, this is where refrigeration is a really big part of reducing food spoilage. Also acid pH, like using vinegar or acetic acid in order to bring the pH down for pickling and that sort of thing can preserve food, prevent it from spoiling, as well as adding salt to it. So these are ways that we have come up with to help reduce food spoilage. Now we want to take a look at the actual chemical requirements. What kind of chemicals are required in order to grow these microorganisms, especially in a lab setting, right? Where we want to grow them under more controlled conditions. So we do see that when we are growing any kind of microorganism, they need a carbon source. The carbon source for chemoheterotrophs are going to be some sort of organic source like carbohydrates, proteins, or lipids like fats. And so they do have to have access to some sort of organic macromolecule. We also see that they're going to need nitrogen. Now, nitrogen is required for them to make proteins, nucleic acids, and even ATP. And so because of this, they're going to have to get nitrogen from something. And we see proteins are one of the best options for nitrogen. You can see the nitrogen here in the amine group on the amino acids, okay, that make up proteins. Another area that we can get nitrogen from are from the nucleic acids. There's always a nitrogenous base present. You can see the little ends in all of these molecules. And remember, we talked about these with nucleic acids when we talked about thymine, adenine, guanine, cytosine. Those are all those nitrogenous bases, and we could get the nitrogen from there. The last one is for the ATP. You can see that nitrogen is present there, and so that is going to be important in creating that adenosine nitrogenous base that makes the A for ATP. Another thing that a lot of these organisms are going to need is sulfur. Sulfur is going to come and be important for some amino acids. So some of our proteins have to have sulfur present in that reference group, that added area, and that's what helps them be able to make those particular amino acids. We also see some vitamins require sulfur. And remember, vitamins are important a lot of times as coenzymes. And so if we need that vitamin in order to make the coenzyme, in order for the metabolic pathway to take place, we need to make sure that we have that sulfur source. Another chemical that is needed is phosphorus. Phosphorus is found with nucleic acids. You can see they have a phosphate group. We see that ATP also has phosphorus. It has 3. That's what the triphosphate means. And we do see phosphorus is important in our plasma membrane. Those, remember, are phospholipids, where the head contains the phosphorus and the 2 tails are the fats or the lipid part. Okay, so these are some chemical requirements that are needed in kind of larger amounts when we're talking about those microorganisms. However, sometimes they need trace elements of some things. So some of these molecules are needed in just very small amounts. These are called trace elements, and they're going to be present in simply using like tap water with a lot of these. The typical role of these, remember, are for cofactors, which are to help enzymes, but they're like iron, copper, zinc, things like that. Now, there's some other essential organic growth factors that might be present for some organisms. If they're essential, they're not provided by their diet, and so they're going to need it in some sort of form. These are going to be a lot of times the coenzymes. That's where the organic part comes from. And so an example of this one that's really important is Haemophilus influenzae. This is a fastidious bacteria. Now, the word fastidious means picky. This bacteria will not just grow on anything, okay? And since it's so picky, it requires 2 molecules that are considered essential organic for it to grow. This includes the X that we see as hemin, which comes from blood, hemoglobin, and the V, which is going to be the NAD molecule. Now, the thing is, you can't just give it blood and be like, okay, it's good, it can grow, okay? Their growth factors are required for their metabolism, so they can't break it down themselves. And so an example here in that red plate is a blood agar. It's full blood cells, but you'll notice there's no bacteria growing on it. And this is because that bacteria can't access those molecules if the blood cell is whole, okay, that it's inside those cells. If we blend up the blood cells and we release all the contents that are inside the blood cells, like the hemoglobin and all that stuff, we see that they can grow. And this is shown in kind of this brownish agar, which is called a chocolate agar. This is where we have actually taken the red blood cells and lysed them open so that the bacteria now can gain access to those special requirements, that X and that V. And so then it can grow. And this is one of the reasons why it's considered a fastidious bacteria, because it is very picky. Now, going back to the metabolism that we looked at in the previous videos, we do see oxygen might be required for some of the bacteria to grow. It's not always the case. And so I have a chart here which shows you the different levels of oxygen that might be needed by a bacteria. So the first one in A here is the obligate aerobes. This means they have to have oxygen. So they grow well on media exposed to the air. They require oxygen as their terminal electron acceptor, and some test positive for cytochrome C because of that electron transport system. But oxygen is required. In B here, we have facultative anaerobes, and these guys can grow with or without oxygen. So you notice here in this little tube, they can grow up where air is present at the top, but they can grow anywhere within the tube as well because oxygen is not required. However, they do grow the best where oxygen is present. You can see that there's a lot more bacteria at the top than there is spread out throughout the broth. This is where these guys are able to switch their metabolic pathways depending on if oxygen is present. All right, a good example of this is E. coli. E. coli is able to do this. It can switch back and forth. The next one are the obligate anaerobes. These guys cannot have oxygen. Oxygen is almost like a poisonous gas to them. And so they need to be where no oxygen is present. They lack the enzymes to detoxify the effects of oxygen. So it becomes detrimental to them. We do see if an obligate anaerobe is to be isolated, we need special collection and we need special transport, because if they get exposed to oxygen for too long, then they will die. And you can see them here in this test tube. They're all growing at the very, very bottom of the test tube. The next ones are the aerotolerant anaerobes. These guys cannot use oxygen, but it's not actually harmful for them. They can be around oxygen, but they just can't use it. They can tolerate its presence, but it's not going to be able to utilize the oxygen. But they do have enzymes that help detoxify the oxygen for them. The last one are the microaerophils. The microaerophils are the ones who only grow with little or low concentrations of oxygen. So you'll notice in the broth, they're kind of in the middle where oxygen concentration is low. It's not zero, but it's low. And they can grow best in that condition. So now that we've looked at the physical and the chemical requirements for growth of microorganisms and bacteria, let's take a look at the media we can use in order to grow them, okay, in the lab. This is known as culture media. The culture media is any substance that we use to grow bacteria on. Now, it could be liquid, and if it's liquid, that's a broth. Or it could be agar, which is like a jelly, a semi-solid. We can put these either in tubes or plates. Now, if it's in a tube, it could be broth, which is liquid. The tube could also be a slant that you see here where the agar is kind of at an angle. And then it can also be where the agar is just deep in the actual tube. We call that a deep. Petri dishes can also be used, but they are only used for agar. We don't use Petri dishes for broths. Now, media must be sterile prior to you inoculating it. So before you add bacteria to it, it needs to be sterile. If something's already growing in it, the liquid is cloudy, or you see colonies of different kind of colors already growing on the plate or the agar, then we have a contamination and we don't want that to occur. So we've got to sterilize these in order to not have endospores or any vegetative elements on them. Once we know that the plate is clean, it doesn't have anything growing on it, and it's sterile, we can do what we call the inoculum. This is what is placed in or on the media to start the growth. And we use a lot of times inoculating loops and even needles and things like that in order to put these organisms either in the broth or in on the plate. Once they start growing, okay, once you've put them on the plate or in the tube and they start growing, we call that a culture. The culture is the bacterial growth in or on the media. And so you can see here in the picture of the broth, the top one is clear. It's got a yellow color, but you can see through it. The bottom picture is still that yellow color, but it's cloudy. This tells you that bacteria is present. If you look at the top plate, it doesn't have anything growing on it, but the bottom plate has all these colonies that are present in orange, yellow, white. Those are different types of bacterial growth. So what kind of media do we need to use? What kind of requirements are needed? And so when we look at agar specifically, all right, that jelly-like substance, there's a couple of things we need to note. That when we heat it up to 100 degrees Celsius, that agar becomes liquid, okay? It gels, or it starts to become that semi-solid at about 40 degrees Celsius. This allows us then to mix the agar and then be able to pour it into tubes or plates to be used. When we make this agar, a lot of times nutrients are added to help the bacteria with their growth. These nutrients would be things like that might have sulfur or phosphorus or nitrogen or carbon in them. They're different things that will help the bacteria grow. So if you look at this picture here, you can see that there are lots of type of media. Now these is probably not all of them that are out there, but each of them has a little bit different characteristics. You can see colors are different. You can see just different characteristics about them. And these are various nutrient augurs and broths used in bacterial cultures. So let's look at the chemicals that end up in a lot of these types of plates and tubes. So agar can either be, or broth can either be chemically defined or complex media. These are going to be for non-selective growth. This is going to support growth of most bacteria that are not picky, that are not fastidious. If you look at the chemically defined, you'll notice that every chemical is defined in the kind of nutrient chart. You'll see that it tells you how much glucose, how much sodium, how much potassium, how much water. It tells you, all right, and it gives you amounts. This is the exact chemical identity of all the ingredients that are known. That means that that agar is chemically defined. If the agar is complex medium, we see that some of the stuff is listed. It contains substances that some of them are listed, but if you'll notice when it says peptones, partially digested protein, it doesn't tell you what kind of protein that is. It then also says beef extract. Well, what's in that beef extract? I don't know. Okay, so this one has where it's giving the stuff it needs, but it's not being as specific. So the exact chemicals, the particular chemicals and their exact amounts are not known here. Okay. All right, and so you can kind of see the difference there. Other media types are going to be things like reduced media. Reduced media is obligate anaerobes can grow on it. Selective media is going to select what gets to grow, so it inhibits some bacteria but encourages other. We're going to see that some of these like encourage the growth of gram-negative but discourage or stop the growth of gram-positive. All right, so it just depends on the type of agar. Some agars are differential, which means they can distinguish between different bacteria based on the colonies that are growing on them. Some media are enriched. These are enrichment medias. This promotes growth of pathogens in low numbers and inhibits others that are present. So these are really big in fecal cultures. So like when somebody turns in a stool sample, there's going to be some bacteria we know will be present in that because it came from your intestines and we know it's there. We don't want to see that bacteria. We want to see if there's anybody who's not supposed to be there. So these enrichment type of media are going to help with that. So they're going to prevent the growth of the normal bacteria that should be there, and we're going to see the growth of the stuff that shouldn't be there. And again, we have a little chart here that shows you the type and the purpose of the culture media. So here are some examples. These are different types of agars that have blood on them. Okay, so they're types of blood agars. The first one is just a nutrient agar with 5% sheep blood. We call this a BAP or a blood agar. This agar is non-selective. Lots of things can grow on it. And you can see here that there's different stuff growing on it. There's like an alpha, okay, like an A. There's the beta with the B, and then there's like the little curlicue. That's going to be our gamma. This is where bacteria grow, but do you notice that they grow differently? Like there's a color that's different, they're showing up different. That's because they use the media in different ways. But this is considered a non-selective type of media, but it is differential on how they use the blood, which is the hemolysis. The second one here is the chocolate agar, CAP. This is where they lake the sheep blood, they blend it up and open it up, and this again is also non-selective. But you're going to be able to grow some fastidious bacteria on these. The last one on here is the Thymomartin or the TM. This is a chocolate agar, but it has also antibiotics added to it. These antibiotics are going to selectively pick what grows and what doesn't. And so it's going to kill off a lot of the different bacteria, and only the ones stronger from those antibiotics are going to grow. This is actually a way to isolate Neisseria gonorrhea. Okay, so this is a type of agar that helps isolate that time. It's selective for it. Some other examples are here. This is the eosin methylene blue or EMB. It's selective for gram-negative bacillus specifically, so gram-negative bacillus species, and it is differential because you can see they grow in different colors. Each one of these is a different type of bacteria. Some grow green, some grow purple, some grow more clear. Okay. We also see a McConkey's auger, okay, MAC. This is also selective for gram-negative bacteria. We also see that it's differential. You can see on one side you have the bacteria in a pinkish color, and the other side it's clear. This is going to be based on what kind of sugar it can use. Is it able to use lactose? So is it a lactose fermenter or is it not? The last one on here is the Hiketonin enteric auger, or the HE. This is also selective for gram-negative bacteria, and it's differential. You can see that you have the black, you have the red, you have where there's no growth, okay, in one of the quadrants. And so it's going to be selective against positive and allow negative to grow. And it's also going to help you differentiate based on chemicals that are present. We also have the phenylethyl alcohol auger, or the P auger, PEA. These are selective for gram-positive. So the last slide was all gram-negative selective. This one is for gram-positive. So you'll notice if it's gram-positive, it grows. If it's gram-negative, it doesn't. Another one is the mantanol salt agar, or MSA. This one has high salt concentrations. And so it's selective for gram-positives that are what we call halotolerant, the ones who actually like the salt concentration. So in this case, you have Staphylococcus, which is showing up in yellow, and then you have Staphylococcus epidermidis, which is on the red side. So then, based on that, you can distinguish the two types of the bacteria. Now, we also see that we have incubation. Incubation may be different based on what kind of bacteria that we're trying to grow. We see that a lot of these can grow just in a basic incubator with a higher temperature that is optimal, that allows them to grow, where it's not too hot. It's like their perfect temperature. But if we're talking about those that need low levels of oxygen or no oxygen, you're going to have special apparatuses to grow those in. And so the capnophils are an example of this. Capnophils grow better at higher levels of carbon dioxide. And they typically, all culture plates are placed in a CO2 incubator. Now there's different types of CO2 incubators. The kind of poor man's version of it, the one that is used in a lot of labs that don't have this high-tech equipment, is that you use a jar, you place your media inside of it with a candle, you light the candle, and it pulls out all the oxygen by releasing also then carbon dioxide from the flame. The flame will eventually go out once the oxygen is gone, okay? And so that's one way you could do it, and that's what we use in our labs. But there are different apparatuses that can also be used in chambers, like you see here in this other picture. These are again for incubation, for especially reduced media, and this would make sure that oxygen levels are not too high. So the ability to grow bacteria is only as good as the specimen collection and the transport that you do. And so this is why it's really important to pay attention to aseptic techniques, which we've talked about in our lab sessions from Unit 1. But it's also important that we select the right kind of media, so the appropriate media for the growth. We also look at the environment it needs to be incubated in, like what's the temperature, CO2 requirements, is it anaerobic, and also the length of time. Some bacteria can reproduce really quick, others it takes longer. And so we need to be looking into those factors. Now, we do have some bacteria that cannot be cultured on these plates like we've seen. These are things like syphilis, Mycobacteria leprae, so like leprosy, cannot be grown on plates. And any obligate intracellular parasites like rickettsia and chlamydia, they cannot grow on plates either. So you have to look at different options when you're diagnosing some of that stuff. We do see that for leprosy, they can grow it on a lot of times armadillos' feet pads, because they like the lower body temperature of the armadillo. That's why leprosy grows better at your extremities, like the tips of your fingers, tips of your toes, places that are a little cooler. But you can't grow it just on artificial media. So how do we obtain a pure culture? How do we make sure that we are only really growing one type of bacteria specifically? Well, to do this, we use a certain method that's called a streak plate method. And in this method, we ultimately hope that we get some colonies that are completely isolated. These colonies are called colony forming units, or CFUs. Now, the way that we would do this is we would take our broth that has the bacteria in it, and we would put a line on our agar plate, okay? That then would be discarded with our cotton-tipped applicator. We then take our inoculating loop and we sterilize it. The sterilizing makes sure that it doesn't have any contamination, and then you run it back and forth against that streak that you just did, okay? Then you're going to sterilize your loop again, okay? Put it in the fire, sterilize it. Once it's cool, you're going to do what this picture shows, where then you're going to pull from the one area and you're going to zigzag back and forth for the two. You're then going to take it and you're going to sterilize it again. You're going to then pull from the two and zigzag for the three, sterilizing when you're done. And then you're going to do it one more time with the fourth, pulling from the third and going in the area in between, making sure you don't go back into one or two, okay? If you go back into one or two, you're going to see that you're going to have poor colony isolation, which is this bottom picture. You don't have a lot of singular colonies present, but the top picture is good colony isolation. You can see that there are colonies that are completely by themselves, not touching anything else. Those are the ones you're going to take from your samples. Okay, so they're really important with that. Now remember, bacteria is going to grow or reproduce through binary fission. So these bacterial cultures are going to have one parent cell that's ultimately put on the media. It will then start to grow into 2 cells, then 4, then 8, then 16, and so on. And it does it through this process of binary fission, and it can actually do it pretty fast. With this, they have to copy their DNA. That's what they do in step 1 here. The cell starts to elongate and it's replicating its DNA. Once it's replicated the DNA, the plasma membrane pitches inward and they create the cell wall in between. Then the cells completely separate. This is binary fission because it does not have a nucleus. We cannot call it mitosis. Now, like I said, sometimes they can do this very quickly. Other times it takes longer. This is known as the generation time. How long does it take for one generation to go to 2, 2 to go to 3, and so on? This varies for organisms. E. coli can do this every 20 minutes. This is why food poisoning can happen within a couple of hours of exposure to E. coli. But tuberculosis, it takes 18 hours for them to reproduce. So it's a lot longer. And so those cultures would need to be incubated longer than the E. coli. Now they do this through a logarithmic type of growth, but eventually they run out of nutrients on the plate. So they will spike in this exponential growth, but eventually they'll stabilize and then start to die because they start to run out of nutrients. There's not unlimited nutrients on these plates. So the generation time determines the culture time and how long you need to incubate it, and also how long it incubates in you before we see symptoms of disease. So if you look here, after 20 generations for E. coli, it went from 1 cell to, look at this, the number of cells is over 1 million. All right, so it can happen really quick. And these 20 generations didn't take that long to get there, okay? And so this is just showing you how quick they could reproduce. All right, so these are the phases of growth. You're going to notice that it's in 4 kind of phases, all right? So this very light green color is called the lag phase. This is the first part, and you'll notice it's kind of like a straight line, okay? It's a straight line. This is a few hours after inoculation. The cells are preparing to grow. So you've put them on the plate, they're preparing to grow. At this point, there's no increase in the population size. Now this is in vitro, so these are on plates. If we're talking about in vivo inside of us, this is also known as the incubation period time after exposure. No symptoms are starting yet. This little bit darker green, the second area, is the log phase. The log phase is logarithmic or exponential growth. You can see that it's taking a very steep upward growth pattern. This is where the cell population is increasing greatly. It's doubling every generation. This is the best time to perform biochemical tests. And so it's important to try to allow this phase to be taking place and then we do our tests on them. We don't want to necessarily do our tests in the later phases because then the cells are older and they won't give us as good of results. Now, in vivo, inside of us, this would be when symptoms of the disease start to appear. Then we have the stationary phase. This is the plateau. This is where the population stabilizes. Death rate and growth rate are equal to each other. So if 2 grow, 2 die, so on. Nutrients are starting to deplete and waste products are starting to accumulate. So conditions for growth are just not as very good now. In vivo, like inside of us, this would be when our immune system starts to attack the bacteria. We start to attack it and fight it. The last phase is the decline phase. This is the death phase. This is where the population is decreasing, and it's decreasing at a logarithmic rate. Okay, nutrients are depleted, death rate's greater than growth rate. And in vivo, this is when your symptoms start to disappear. We call this convalescence. That's what starts to begin. Now, this does not mean you're not contagious still. It just means you don't have as many symptoms. Okay, so this is growth, of course, in vitro, but we do see that it gives you a little note on what happens if it's in vivo in a live culture. All right, so be aware of these. You need to make sure that you understand these 4 steps because you're going to be asked about them on an exam. Last but not least, we want to talk a little bit about methods of measuring microbial growth. How do we measure it in the lab to see like how much bacteria is actually present? We can do direct type of measurements, and these are quantitative where we're looking at the numbers. We're actually looking at plate counts here of CFUs, and we do this with serial dilutions because if you look at the first plate over here, there's so much growth you can't count that all. And so what we do is we use this technique on food and environmental specimens to determine the bacterial level of contamination, but we have to dilute it. So we start with our first plate and then we dilute it and we dilute it and we dilute it until finally you can count 3 on the last plate. There's 3 colonies. However, it's not just 3 colonies. It's where it's 1 to 100,000. So if there's 3, that means there's 300,000 bacteria technically there. All right. So you must multiply the number of CFUs by the dilution factor in order to find out exactly how many are present. These are usually reported in number of CFUs per milliliter. Okay. Now, in our lab, we will use a little bit of quantitative bacteria growth on urine cultures, but we are going to use it by doing a calibrated loop. These calibrated loops are super small. They only collect 0.001 milliliters of urine, so a very small amount. And it allows us to then take it and do our culture, so a zigzag method, so that we only potentially have a few colonies to count. The thing is, though, because of this dilution, okay, since it's 0.001 milliliters, this is going to be 1,000 times. So just like we saw before, you count how many colonies. There's 8 here. You do 8 times 1,000, so there's 800 or 8,000. All right, so that's what you're looking at here. Okay. A more indirect way to look at the amount of growth is by looking at it semi-quantitatively, where we look at turbidity. How much light can get through? How cloudy is the broth or liquid? The more cloudy it is, the more bacteria that is present. Now, you can use a spectrophotometer for this. Spectrophotometers are going to give you the actual amount of light that's going through. And so it can give you a number. That's why it's kind of semi-quantitative. On the other hand, you can also do the McFarland's standard, which is where you use this card that has these dark, bold lines on it. And you place it behind your tubes and you see how well you can see through them. Okay, and so this is a type of Kirby-Bauer antibiotic sensitivity test. It allows us to see how sensitive the bacteria is to the different levels of antibiotics. In the lab too, we will use the semi-quantitative measuring of bacteria where we'll kind of gauge it. Is it light growth? Is it moderate growth? Or is it heavy growth? I'm not going to make you count each of the colonies. But obviously, if it's too many to count, like you see here with the yellow bacteria, that's heavy growth. I'm not going to make you count that. You could count colonies, and we could do semi-quantitative by putting them in quadrants. If you look at this bottom red one, it has like the 4 quadrants. You could count one of them and then multiply it by 4 to give you kind of a idea, an estimate of how many colonies are there. But again, this is going to be semi-quantitative. Okay, so it's not going to be giving you all of it. All right, so this finishes up looking at microbial growth factors. So the next one we're going to look at is how do we control this now? How do we control how the bacteria is going to grow in different situations? So if you have any questions, please let me know.